Hello, everybody. Welcome to this week's webinar on how to explore metabolic pathways in VEuPathDB. Today, the webinar will be presented by David Roos from the University of Pennsylvania, myself, Evelina Basenko, located at the University of Liverpool, and we also have members of staff that will be answering your questions in the background. So while we wait for other people to join us, I would like to ask you to fill out a poll. I'm posting a link in the chat that you should be able to see now, and also shown on my screen currently. We would love to know which database or databases you use on a regular day basis, and I will display the poll results live, so we'll be able to see your responses in real time. So I see there's some fungal folks, ToxoDBs coming. Excellent. So to those that just joined us, welcome. We are running a live poll on which database or databases you use in your research, and the link to the poll is at the top of the screen and also in the chat. So click on the link and let us know what is your preferred resource. So also just a few organizational things before we start. The webinar is being recorded, so if your colleague was not able to make it, or if you have to leave early, you can always catch up later. We will post the link to the recording on our website, YouTube channel, and it will be also sent out in the follow-up email from the GoToWebinar panel. That will also include certificates. So if you are interested in receiving a certificate for attendance, it should be in your inbox in a day or two. And if you don't get it by Monday, let us know. As far as the questions, if you have questions, please type them in the chat in the questions section, and we have several members of the outreach join us, so we'll try to answer them, and time permitting, we will also try to answer them during the webinar. Okay, so I see a good number of people joined. Thank you very much for providing your answers. So I think we're gonna go ahead and start, and I will navigate to FungyDB first to give you a brief introduction into the beta site before we move on with our demonstrations. So if you haven't seen the beta site yet, this is the new look. The site has several main components, such as the banner at the top that also has a series of useful links that I will go over quickly just in a few seconds. We also have a panel on the left, and these are specialized searches that allow you to create in silico experiments. We additionally display specialized searches access at the top here, and that's because if you navigate from the main page while creating strategies, this banner will be always with you, so you can always come back and initiate a new search if you like. We also have a series of various resources and tools designed to get you started and learn about various types of data we have and how to mine it. For example, if you would like to learn how to search transcriptomics data set, there's a quick card that provides more information about what type of data we have and how to access it or view it. In addition, we have news and tweets section on the left that can be expanded or collapsed. And in the news section, we post updates about various data sets uploaded, new tools, etc. And follow us on Twitter or Facebook, and you can also catch up with posts here online. The main menu at the top is located within the banner and under the site search menu provides quick access to various types of sections. First, you can access your strategies. And while I am signed in currently in FunGDB, you don't have to have an account to mine the data. You can create any searches that we will demo today without having an account. However, if you would like to save the strategy or share it with others, you'll have to create a free account, which is really easy. It takes just a few seconds so that you can generate a unique URL to that strategy. Furthermore, you can access specialized searches, as I have mentioned, initiate various searches for genes using these type of criteria, and investigate other types of data, such as metabolic pathways that we'll take a look today, and also compound records that can be searchable based on the gene IDs, metabolic pathway names, etc. In addition, we have a tools section that allows you to access various tools available in our resources, including Galaxy, private My Workspace. This is where you can analyze your data imported from Galaxy using the FunGDB or VEuPathDB interface. You can also look through the types of data we have, gain more information about us and how to submit data for integration in VEuPathDB sites, and get help. When I mentioned that we'll be posting the link to today's recording, this is where you would find it if you click on the Learn How to Use VEuPathDB databases. We have a series of sections. One of them is webinars. So if you click on the Explore, it will take you to the page that lists all previous webinars, also upcoming webinars. And today's webinar, once you navigate to it, will have additional details with the link included. And finally, before we move on with demonstrations, if you ever have questions or suggestions, feel free to contact us. Click on the Contact Us link to submit a form, and we'll try to get back to you within 24 to 48 hours. And I think I would like to point out that you can also follow us on Twitter if you click on the social media links. And with this, I will transfer my screen to David Bruce, who will take you through the background information on metabolic pathways and also show you how to create searches. David, if you're talking, you're muted. I wonder if we have some connection issues. Can anyone hear me?
I can hear you, Eve.
Okay.
What I see is a blank screen with the announcement Metabolic Pathways in VEuPathDB.
Okay.
Right. Perhaps we should move forward to what you were going to demonstrate, and I'll see if I can contact David.
Sure, okay. Sounds good. All right, folks, we have technical difficulties, but that's okay. So I will go back to FunGDB, and we'll take you through a search strategy that I think will demonstrate how you can create not only various search strategies, but how to mine data when it comes to metabolic pathways. And let me just become a presenter really quick again so I can share my screen.
Sorry, Eve.
Yes?
I think I am back again. Very sorry for that. GoToMeeting webinar sent me a panel that I couldn't see behind that wouldn't let me access again. I'm ready to get started if that's all right with you.
All right, no problem.
And very sorry all for the technical difficulties. There's a new version of GoToMeeting webinar that didn't like my laptop for a moment. In any case, thank you, Eve, for the introduction and welcome. We will be talking today about metabolic pathways using a couple of examples. As you can see on my screen, an example from TaxoDB, but we will also use examples from FunGDB and from Vectorbase, and of course, as is always the case, this uses a common infrastructure. So the tools that we're displaying should be illustrative of what you can look at for whatever organisms you may be interested in, for trypanosomes or microsporidia or giardia or entamoeba or whatever. I'll also point out that we are using the newly merged database, in this case the beta site for TaxoDB, and I would encourage all of you to use these beta sites, which will become the default view when we go live with the new release in a week's time or so. And that will be true for all of our sites, but it will also be true that you can always access the legacy sites that you are used to. What I thought I would do today is start by looking at just an individual gene, and it's an individual gene that is a metabolic enzyme, since we're talking about metabolic pathways today, and use the gene page as a launching point to look at some of the other more specialized and informative views that allow us to see how this particular enzyme fits into metabolic pathways, and to discuss how those assignments are made. This particular enzyme, deoxycellulose phosphate reductoisomerase, is associated with the apicoplast, a parasite-specific organelle co-opted from an ancestral alga in many but not all apicomplexant parasites, and an appealing drug target, for example, the target for doxycycline or clindamycin or spiromycin, which are clinically used for treatment of toxoplasmosis and malaria. I'll drag over just for a moment the original metabolic pathway view from the original Plasmodium genome paper, highlighting the importance of the apicoplast, including the DOX-P pathway for isopronoid biosynthesis that is attractive as a drug target because it's distinct from the HMG-CoA reductase pathway used by human hosts. That pathway has been mapped out in much greater detail here, for example, from a review paper by Stuart Ralph. And we know these pathways are essential from studies that have been published by Syntony and Boris Streepen's lab, and in Plasmodium from work on Ellen Ye, who's shown that you can rescue the pathway by supplementing with the downstream end isopentenyl pyrophosphate. So an interesting pathway for sure, and one that we can use as the basis for some of our analyses. Looking at this individual gene, as I scroll down the gene page, which should, I trust, be familiar to most of you, we can navigate through the menu over on the left to information about gene models and taxonomy and functional analysis. We see that the gene model itself is highly supported by RNA-seq evidence, so we're pretty confident in the structure of this model. I should point out that despite the extensive literature that's been published on this, there are no user comments that have been added to the gene. So I went in last night and added a couple of user comments, and I would encourage the rest of you to do the same for any gene that you may find of interest. I've added some of those key references to the user comments page, and added some synonymous names. This name, deoxycellulose 5'-phosphate reductoisomerase, is also known as DOX-P, DOX-PR, DOX-P reductoisomerase, DXR, ISPC. These are terms that will allow you to identify these. At the moment, those user comments have been added in the legacy site, as you can see from the more old-fashioned banner. But once the sites merge, this will be available, carried forward in the more modern site that we are working on moving forward. Scrolling down the list, we can see further information about taxonomy, orthology, and Syntony. And the orthology view allows me to highlight how we know the information about this gene, even in the absence of formal curation, which has not captured the information from the literature. We can see that this gene and its structure are present in multiple Toxoplasma strains. And if I enable scrolling and zooming, I can scroll down to see that the same gene is present in the same location. That is, it's a synthetic gene in other related species as well, and Homondia and Neurospora and Sarcocystis and Neurospora. And we can get further information on that by looking at the orthologs that are present. I will navigate over to OrthoMCL by clicking on this link, and by navigating to OrthoMCL, I can see the phyletic pattern map that you may remember from the OrthoMCL webinar for those who attended that or would like to look it up on YouTube. By looking across all of life at a whole variety of species, I immediately noticed that this particular gene is present in most but not all apicomplexan species, highlighted in magenta. In this case, looking at a Toxoplasma strain, we can see one such enzyme. It is absent from virtually all of the fungi, metazoa, many other eukaryotes, but present in plant taxa and bacteria, but not archaea. This is a bewildering list, but it certainly meets the criteria that we would expect for an organelle that was horizontally transferred from the plastid of an ancestral alga. We can see that the domains associated with that protein include several well-conserved PFAM domains that are associated with this enzyme. And as we scroll down across all of life, we can see a consistent representation in most of the species that are represented here. I particularly like the cluster graph, view, which allows us to look at the relatedness of the various taxa that have been identified. The colors specify what these are. If I want to ask about Plasmodium, for example, I can see that Plasmodium falciparum shows up here in this cluster. And indeed, all of these are Plasmodium that are quite closely related, somewhat distant from, but still significantly similar to Toxoplasma isolates and various plant and bacterial taxa that are indicated here. The surrounding color corresponds to the same color that was shown on the pattern map we'd seen before. A lot of information on this slide, but what we would like to do is to try to gain some functional insights as to whether these are indeed the same enzymes. And we can do so by painting this pathway map, by painting this orthology file with PFAM domains. And we can see, as we've already seen in the map before, that the same pattern of sequence motifs that defines this ortholog group is conserved across virtually all of the isolates, a few outliers shown up here. Or we can paint that by EC numbers, and this allows us to see that this particular EC number, an enzyme commission number represented by 4 increasingly specific digits, 1.1.1.267, which defines in the first instance that this is an oxidoreductase, in the second instance that it acts on a hydroxyl group, in the third that its acceptor is NAD or NADP, and the fourth, the enzyme specific name. We can see that many, but not all of the genes in this ortholog group are annotated with this specific EC number. And what that allows us to do is to transitively apply as a putative in silico EC number assignment, this EC number to the other members of this group. That's not something we could do if there were multiple EC numbers that were identified here, but we have pretty high confidence that all of these should be annotated with the same with the same EC number. So let's go back to our view here. We know something about the ortholog group. We know from phenotypic information, in this case genome-wide CRISPR score is carried out by the Laredo lab, that this particular gene shows a high degree of essentiality, a fitness score that is where a knockdown is down 2 to the 4th fold, 16-fold in its fitness. We could examine variation data, as you've seen in previous webinars, transcriptional profiling data from a whole variety of studies, summarized at the top, indicating the levels of information. And I'll scroll past this information, although we will come back to it later on, simply to point out that there are differences between in experiments. For example, when we look at an experiment that highlights oocyst differentiation in Toxoplasma, we see that unsporulated oocysts on day 0 express virtually none of this transcript, but transcription is increased significantly by day 10 of sporulation as parasite oocysts progress through their cycle. We'll come back to some of these others in a moment. Scrolling down towards the bottom of the page, further towards the bottom of the page, we can see the features that we identified in OrthoMCL, PFAM domains, and other interpro domains mapped here onto the genome. Functional predictions include protein targeting and localization. Here, mass spectrometry carried out using the low-pit technique, in this case data from Ross Waller's lab, highlights the profile in subcellular fractionation combined with tandem mass spec to predict the subcellular localization for this enzyme. And in this case, we can see that it is correctly assigned to the apicoplast. And finally, EC numbers that have been assigned to this, the EC number 1.1.1.267, available by formal curation in GenBank, but also computationally inferred from Orthology, as we've seen in OrthoMCL, Go terms, and so on. What pathways is this associated with? We're focused on metabolic pathways in this webinar today, and we can see that this enzyme is associated with a bewildering number of pathways, in this case pathways culled from both the biopsych, the metapsych pathways, and from KEGG pathways as well. So that highlights some potentially exciting information. For example, I didn't know that this enzyme is associated with the degradation of chlorocyclohexane, an important industrial contaminant. I wouldn't have thought that toxoplasma could be used for bioremediation. So let's jump in and take a look at that metabolic pathway. By clicking on that, it immediately loads a metabolic pathway, which is represented here in Cytoscape, and we can see the metabolic pathway. Using my trackpad, I can zoom in to see individual enzymes and metabolites, or zoom out to see the entire pathway. And the enzyme that we're interested in here is highlighted in yellow. This enzyme with the EC number 1.1.1, some sort of oxidoreductase without the specific enzyme. This suggests to me the enzyme in question, DOXP reductoisomerase, could potentially be used in this pathway, but you'll notice that all of the other steps on this pathway are missing. So despite the fact that I was initially excited to see hexachlorocyclohexane degradation as associated with the enzyme I'm interested in fact, I can see that it's only this class of enzymes that might be involved in this pathway, and the absence of toxoplasma orthologs for the rest of the pathway strongly argues that this metabolic pathway is not biologically active or relevant in toxoplasma. I can sort these columns in various ways by the EC numbers, by the numbers of reaction. I'm only going to be interested in those that match the entire EC number place. So I have filtered over here on the right, or sorted over here by clicking on the column at the right to those that show yes. And I can see a much more limited number of metapsych pathways or KEGG pathways that include the full enzyme in all of its glory, all four of the subcomponents of the EC number. So let's look at what we would expect to see, the isopronoid biosynthetic pathway. And when I click on the isopronoid biochemical pathway here, this will now, where did that, there we are. This pathway now came up for me, and I can see something that looks very different than what we saw before. An entire pathway all the way from the substrates of glyceraldehyde 3-phosphate and pyruvate down to the isopronoid unit that is produced. And this pathway is much more heavily populated with enzymes that are present in toxoplasma, as represented by the yellow boxes. Not every pathway, not every enzyme is present. For example, this enzyme here, the 1, 2, 3, 4, 5, 6th step in the pathway, I think this is the ISPG enzyme, appears to be missing from toxoplasma. That's surprising if this pathway is accurate, or if indeed the metabolic pathway maps that I showed you from publications are accurate. And I can explore this a little bit further by, for example, clicking over to OrthoMCL again to ask about ortholog groups with this EC number, and that identifies exactly one ortholog group, which shows the same phyletic pattern that we've seen before, present in most, but not all apicomplexa, present in plants, present in bacteria, absence in humans, mammals, other metazoa, absent from the archaebacteria, distinctive PFAM domains, and a cluster graph that I have painted with EC numbers now to show a slightly different pattern than we'd seen previously. We can see that the bacterial taxa here are characterized by one particular EC, are annotated with one EC number, whereas the plant and parasite pathways are annotated with another EC number, 1.17.7.1. This is identified by OrthoMCL as being part of the same ortholog group, presumably carries out the same function, which simply was not recognized by the KEGG pathway because the KEGG pathway only included this EC number with a terminal 3. Now, you'll recall that there were several pathways that showed an exact match to the EC numbers here. Turning, for example, to look at metapsych pathways for isopronoid biosynthesis, we can see that the metapsych family of databases identifies sorry, that was metapsych we were looking at before. Let's take a look at the KEGG pathways. If we look at the KEGG pathways involved in terpenoid biosynthesis, we see a slightly different pattern. Sorry, okay. Let's see. We see a slightly different pattern of these enzymes, in which the same pathway that we looked at previously is indicated over on the right here, missing this one step. But the KEGG pathways are more fully annotated rather than being driven by E. coli. And now we can see the presence of this enzyme, which, yes, is indeed the enzyme that we suspected that is there. So this pathway map highlights to us that that toxoplasma has all of the enzymes we would predict would be required for taking pyruvate and glyceraldehyde 3-phosphate down to isopentenyl diphosphate, or a prenylated protein here. This slightly larger zoomed out view of intermediary metabolism also shows us alternative pathways. This on the right is the mevalonite pathway, which humans use to synthesize isopronoids and sterols. And there's no evidence of these pathways in toxoplasma. We can explore this a little bit further by painting this metabolic pathway map by individual genera. So I'm going to paint on top of this all of the apicomplexa, and just for kicks, human pathways as well. And as we paint this on top, this phyletic pathway information, the ortholog information that we've already explored on the gene page and in OrthoMCL, is now dynamically in real time being queried and pulled up onto this pathway page, and confirming what we have already discussed. Confirming, for example, that this enzyme, which, yes, is DOXP reductoisomerase, is present in coccidian parasites, Eimeria, Neospora, Toxoplasma, present in Plasmodium, present in the pyroplasms, Babesia and Theileria, absent from Cryptosporidium. A surprise that was discovered some years ago as the genome for these organisms was analyzed. Cryptosporidium, in the course of its history, has lost the apicoplast and consequently the enzymes associated with it. I didn't know before I looked at this last night that Gregorines also appear not to have any DOXP reductoisomerase, or indeed any of the enzymes along this pathway, lending confidence in this assertion. And of course, humans also lack this xylulose pathway for isopronoid biosynthesis, but contain the enzymes associated with the DOXP, with the mevalonate pathway, such as phosphomevalonate kinase. Indeed, we can find any information on this from the functional studies that you're already familiar with. Let's paint on, for example, the information that we already looked at from functional expression. Let's see if I can find the OAssist data that we looked at before. Here is the OAssist RNA-seq data. I'll paint that OAssist RNA-seq data. This is the graph that we looked at earlier that highlights the increased expression of this transcript for DOXP reductoisomerase with differentiation of OAssists. And I can now see that across the entire pathway, there is a strong trend where all of these enzymes are more heavily expressed in the replicative forms as the parasite forms mature sporozoites during sporulation of the oocyst. And of course, we have available to us any of the information that is available in VEuPathDB databases if we wanted to look at a wider range, for example, of transcriptional profiling studies across a broader range of the life cycle. Let's take a look at the development of sexual stages, paint this pathway, and now we see a different pattern showing the moderate levels of expression in tachyzoites, low levels of expression in the latent tissue cyst forms, higher levels of expression during the differentiating sexual stages in feline enterocytes. So this is a very powerful functionality that you can use to explore any of the metabolic pathways that you may be interested in for any of the organisms. Let's finally go to take a look at a metabolic pathway query. In this case, I have sought genes that are associated with the apicoplast by virtue of that proteomics assessment, the low pit analysis that we described earlier, that identifies a list of 148 genes, which I can explore here or analyze further using the tabs at the top of the result page that many of you have seen previously, looking at the mapping of these individual genes to the genome. I can see that these genes are distributed throughout the various chromosomes. This will take a little while to load. Maybe we'll come back to it in a moment. But I can also configure my queries by analyzing results to look at enrichment in Go terms or enrichment in metabolic pathways. When I enrich for Go terms, an analysis that I ran previously, it identifies pathways that are significantly enriched here, the fold enrichment and multiple testing correction scores and p-values for fatty acid synthase activity, oxidoreductase activity, pyruvate dehydrogenase, various activities known to be associated with the apicoplast. And similarly, I could look for an enrichment in metabolic pathways. And by running an analysis of metabolic pathways, I identify enrichment in a variety of different pathways, fatty acids for biosynthesis, for example. This biosynthesis of antibiotics is a broad scale overview of intermediary metabolism, which encompasses many of the pathways that we might be interested in. As this comes up, I think you'll be able to see, or perhaps I have this preloaded. This is a large pathway and is going to take a little bit of time to load. So what I'm going to do is maybe turn this back over to Eve to show you some examples of queries in FungiDB. And if there is time, I'll come back to show a little bit of further analysis in a vector species. Thanks very much, Eve. And please remember to feel free to type any questions that you may have into the questions window.
All right, thank you, David. Yeah, all right, you should be seeing my screen now. All right, so we'll switch gears and I'll show you just an example of searches and also visualizations that you can do in FungyDB. So let's just imagine that I'm an aspergillus researcher and I'm interested in general how cells respond once they invade the host. And in this particular case, I will show you an example with Aspergillus fumigatus. We know it's a fungal pathogen that causes invasive pulmonary disease in immunocompromised hosts, and it's quite tricky to treat, especially when it establishes a system of filamentous hyphae inside tissues. So I'm a researcher that interested in glycolysis in general, and I read a paper recently, and I looked at a few genes that kind of look cool when it comes to upregulation when cells were incubated either with blood media or that were subjected to hypoxia. And so I searched for 4 genes, and I won't go through every step of setting up the query. If you would like to learn more about how to create this type of queries, we have previous webinars, but I will go through individual steps that I have involved to build it. And so the 4 genes that I have in mind, I used the gene IDs search to identify them, and I essentially navigated to searches, genes, annotation, gene IDs to deploy that search. And then when my search returned the genes of my interest, I then asked the question, well, how many of these genes actually are upregulated when cells are exposed to hypoxic condition? And in this case, I have cross-referenced the 5 genes that I'm interested in with the microarray data. And the microarray data came from the experiment where cells were exposed to hypoxia for 2, 6, 12, and 24 hours. Samples were collected, and they were compared to the 0-hour control sample. And so my setup is actually looking for any genes that have a gene expression between 60th to 100th percentile for any of the genes on the list. And when I ran the search, I identified that 4 out of 5 genes that I had actually fit that criteria. They're upregulated during hypoxia. And if we take a look, if we take a look at the list of genes that I have, they match glycolysis pathways. So, for example, here is a dehydrogenase and glyceride 3-phosphate dehydrogenase, and I think this one, TFP, right? The isomerase is also involved in one of the glycolysis steps. So far, right, what I have looked for, this matches the genes that I'm interested in. And the next step what you could do, you could look at individual gene record pages, just as David showed you before, and investigate the individual components of that page for metabolic pathways, EC numbers, to try to figure out what information is available. But if you have a list of genes and you just want to transform them into the pathways and see what comes up, you can add a step. And adding a step is sort of an essential step of building a strategy. Imagine that you could expand the strategy to look at SNPs, RNA-seq data, phenotypes, anything. In this particular step, I'll click on the Add Step button and select an option that will transform the genes that I have into metabolic pathways. And when I click on the option, I'll be presented with a screen that allows you to set up the parameters of your search. So we have 2 options to essentially, you can use any of the pathway sources, or unless you like to select specific one, I'll leave this at default as well as the EC numbers. However, I would like to run a search where I'm exclude any incomplete EC numbers to decrease the number of hits that are with sort of less less believable in a sense, because you may have a lot of genes that have non-extensive EC annotations, and we would like to eliminate those from the results. And so the 4 genes are mapped now to various pathways, and as you can imagine, glycolysis will be one of those returned. And glycolysis is quite a large pathway that also has additional pathways fitting in. And so let's just take a look at this particular one, and I'll open it in a tab. And we'll see how else you can explore not only the data for Aspergillus, but also for other species. So this is the screen that you are already familiar with, and something Dave showed you. You have general banner that gives you little bits of information about the pathway itself. And if you scroll down, this is the visual area that can be also adjusted. So you have a number of controls on the left, and obviously an option to drag your screen if you like. You can zoom in, zoom out, and I'll show you a few things that you can do within this layout. So let's go back to my strategies and actually see if we can find one of the enzymes on my list. So the 4 genes that were upregulated during hypoxic conditions have relation to the glycolysis, and this one, glycerate 3-phosphate dehydrogenase, is actually an enzyme that catalyzes the 6th step of the glycolysis. In addition to that, it also has important function in non-metabolic pathways, and that includes transcriptional activation, apoptosis, and other functions. So let's take a look at that enzyme. I'll come back to the metabolic display and zoom in on the 6th step and click on the node. So this is the glycerate 3-phosphate dehydrogenase enzyme that is also linked to the gene ID that I just shown you in the previous screen. So you notice that you can reposition if you would like to sort of sort out the location-wise various nodes. You can zoom in, zoom out, and you can also save the screen that you see as a JPEG or a PNG. To save the screen that you currently see, you can click on the file, save it as PNG and JPEG, and it will be exported. Obviously, you can return to the initial view if this is a smaller pathway and export images as such. We also offer a way to export Json files, and this is maybe useful for those that would like to export the pathways networks into a web browser. So you can download it as well and work with it locally. So in addition, let's return actually to our enzyme and click on it again. The next step that I would like to do is paint the nodes based on the data used from the experiments that are listed in FungiDB. So data shows you that the experiments that you can use are not necessarily attached to particular species. We have looked at the records in Aspergillus fumigatus using a list of gene IDs and then microarray data. But to paint the data, you can use any of the data sets that are currently integrated in the VEuPathDB sites. For example, here's a data set that I have investigated in my search strategy. So I can click to select this data set and say paint. And notice that once the page refreshes, you'll see all of the data that are available from this data set being painted over individual nodes, and these are the genes that are related to these nodes. So you can essentially take a very broad view at what is happening within that or another pathway on the global scale when cells are assayed at these conditions. And we can click on the node now, and once the pop-up window refreshes, you'll see that the individual samples will be displayed here on the bottom. Here we are. And so now you can see not only how the data are mapped throughout the pathway, but also how individual samples fit in within this node. So where things are being upregulated, downregulated, etc. What is also really cool about this particular view that you can cross the barriers of species, right? You can select experiments that are outside of your current strategy. And for example, I may be interested in looking at Candida auris data, Candida albicans data that we have recently integrated, where cells had been exposed to macrophages, and then the RNA-seq data were collected for various from various samples. So when I click paint, this is a little bit more complicated experience, so it may take a little bit longer to load, but I could click on the window again to refresh the node details, and this time I will be able to visualize essentially every single sample from which we have data for this node for Candida albicans. And so this may be a little bit hard to see, but we have several samples available from this data set, and that's including infected macrophages that have live Candida cells, infected macrophages with dead cells, and then also samples that can be used as control, such as when Candida albicans has been grown without macrophages and also unexposed to macrophages. So what is interesting is that it looks like after the initial 0 hours time frame, then you have a drop down in the expression of those genes and where the live Candida are actually engulfed by macrophages, and that is consistent with what we know about glycolysis and host cell interactions from other species. For example, we know that cells that are actually engulfed by macrophages and undergo carbon starvation activate glyoxylate cycle, while those that remain sort of unexposed and free of macrophages will continue to access the glucose, and as such the glycolysis cycle will be active. So that's pretty cool. But in addition to this node, you can essentially examine the data for various nodes very quickly without running dedicated searches within the search strategy. So I think this is all that I wanted to show. So David, I will pass it back to you unless there are any questions.
All right, thank you very much, Eve. Let me just show my screen once again. And let me make sure that we have the right screen. All right, can you see my screen?
Yes.
Fine. So let's return to where we were before. I had run a query asking for genes predicted to be associated with the apicoplast, not based on their sequence, not based on curation, but based on their subcellular, predicted subcellular localization from low-pit mass spectrometry.
David?
Yes?
I'm sorry to interrupt, but what we see is many open windows.
Okay, that is...
I think you just want to show the one that was on the right.
I'm not quite sure yet. Let's stop sharing that screen and try to share the proper screen again. No. Okay, sorry about that. Try this one. Okay. There we are. I believe now you're seeing just the screen that we wanted to look at. So yes. We took a look at that set of genes that were associated with the that are predicted to be associated with the apicoplast from this localization, and the localization of those across the genome, which is was loading slowly, and a gene ontology enrichment that I had on previously that that made sense in terms of various pathways that we could look at, but also at metabolic pathway enrichment. The biosynthesis of antibiotics, as I mentioned, is a large pathway, and it represents most of intermediary metabolism, and it's not surprising that this enzyme is particularly enriched in intermediary metabolism as a whole. But we can also look at the other pathways that might be associated with that. So in order to do so, let's switch over just for a change of pace to run a similar query against a vector species. I have previously run a query to identify genes from a high-quality data set recently released of specific tissues associated with sensory perception in Aedes albopictus. And in this particular case, I configured the query to ask for genes that were upregulated at least tenfold in Aedes albopictus antennae, as opposed to the whole body transcriptome of female Aedes albopictus genes. And these genes, again, we can analyze this list of 798 genes. We can analyze in various ways. We can see that they are distributed throughout the contigs of the assembled genome. No obvious clustering there. But we can also analyze these to look for enrichment on the basis of various kinds of terms. We can, as you have seen previously, we can look at enrichment of the text terms associated with the annotation of those 798 genes. And as that analysis is run, we can see the not very surprising result that genes upregulated in antennae as opposed to the entire organism are dramatically upregulated in odorant receptors, as one would certainly expect. But we can also ask about the enrichment of those. We can also ask about enrichment for Go terms, or for example, for metabolic pathways. I'm going to run this using the default criteria to ask about which pathways may be most strikingly upregulated. And we see nothing like the level of upregulation seen for odorant receptors, but certainly a suggestion of enrichment in these pathways associated with carbapenem and chloroalkane metabolism, glutathione metabolism, intermediary metabolism of a number of toxins and metabolites that the mosquito might encounter. And we can zoom in to take a look at those, as we have already seen for Toxoplasma and for Aspergillus, looking, for example, at the pathway associated with Carbonyl Biosynthesis. And as we have looked at previously, we can scroll down to take a look at this pathway and see that this pathway in all of its glory includes enzymes which are highly represented throughout that are identified in the Aedes albopictus genome. And we could paint this in various ways that we might be interested in, looking, for example, at the transcriptional profiling for the genes that I was looking at, the sensorial organs in Aedes albopictus, and those pathways will come up slowly, replacing the yellow symbols indicating the presence of a gene with the transcriptional profiling map that we've seen for these individual enzymes. Clicking on this will show us what the pathway map recognizes. So that's all that I wanted to talk about today in terms of metabolic pathways. Just to review, we have seen metabolic pathways and I've seen enzyme names that are associated with individual genes, metabolic pathways that one might want to explore. We've talked about how those metabolic pathways are, those enzyme IDs are assigned by Orthology, how we can paint these pathways with functional information of interest, and how we can extract and how we can extract that in a variety of different organisms. Since we have a moment or two remaining, let me try to modify this a little bit. We looked at these individual scriptome genes. We also looked at the metabolic pathway enrichment, but we can also ask about all of the pathways associated with all 798 genes that we might be interested in by transforming that list of genes into specific ortholog groups, compounds, or metabolic pathways. And I'm going to ask for pathways that come from either Metapsych or KEGG that contain any of these genes, insisting on an exact match and only for full 4-term EC numbers. And that search will identify all of the pathways associated with this set of genes, which in this case I identified as upregulated in antennae, but which you might wish to identify based on your own analyses of genetic mapping or subcellular localization or so on. All right, let me leave it at that. Ask if Suzanne or Eve have any other comments that they wanted to add, or if there are any questions that have come in through the chat window that we'd like to discuss.
Nothing here, and no questions from the audience.
All right. Maybe back to you, Eve, to close us out for the day.
All right, sounds good. Thanks so much, David. All right, I think we will wrap this up. Again, if you have questions or if you can't figure out how to set up any strategies or how the data is being integrated, just click on the Contact Us link and send us an email, and we will be happy to answer any of your questions. And I think that's probably will be it for today. So thank you everyone for joining, and hopefully we'll see you soon. Bye.